Hello guys, today I will be talking about the composition and function of fish blood and I will try to relate it with humans. I hope you will like the lecture and give your valuable comments in the comments box. Your suggestions will definitely help us to improve. Blood in both humans and fish is composed of plasma and formed elements. Plasma is a yellowish liquid that carries nutrients, hormones, and waste products. Blood performs several essential functions in the body, including transport of oxygen and nutrients, removal of waste products. Blood also carries waste products, such as carbon dioxide and urea, away from the body's tissues and organs to be eliminated. Regulation of body temperature. Defense against infections. White blood cells in the blood help to fight off infections and diseases by identifying and attacking foreign pathogens. Clotting. Maintenance of acid-base balance. Blood helps to maintain the body's acid-base balance by regulating the pH levels of body fluids. Hormone transport. Blood carries hormones and other signaling molecules throughout the body, helping to coordinate and regulate various physiological processes. Blood makes up 6-8% of total body weight in both humans and fish. Blood is composed of plasma and formed elements. Plasma is a yellowish liquid that makes up about 55% of the blood volume. It is mostly water, about 90%, and contains various proteins such as albumin, globulins, and fibrinogen, as well as electrolytes, hormones, nutrients, waste products, and gases. Formed elements are the blood cells and platelets, which make up the remaining 45% of the blood volume. In humans, blood cells are continuously formed in the bone marrow through hematopoiesis. Hematopoietic stem cells give rise to different types of blood cells, including red blood cells, white blood cells, and platelets, through a process of differentiation. These cells are released into the bloodstream, where they perform their specific functions to maintain the body's homeostasis. The process of blood cell formation is regulated by hormones and growth factors and occurs throughout a person's life. In contrast, blood formation in fish occurs primarily in the kidney, spleen, and specialized blood-forming tissues called hematopoietic tissues, which are located in various parts of the body, such as the liver and the thymus gland. In some fish species, the head kidney, an organ equivalent to the mammalian bone marrow, plays a crucial role in the production of blood. As discussed earlier, blood is composed of plasma and formed elements. Plasma is a yellowish liquid that makes up about 55% of the blood volume, and it contains various proteins, electrolytes, hormones, nutrients, waste products, and gases. The formed elements include red blood cells, white blood cells, and platelets, which make up the remaining 45% of the blood volume. The red blood cells carry oxygen, the white blood cells help in the immune response, and the platelets aid in blood. RBCs in fish are similar in function to those in humans and other vertebrates, but they have some unique features. Unlike mammalian RBCs, which are anucleate, lack a nucleus, fish RBCs are nucleated and contain a different type of hemoglobin that allows them to extract oxygen more efficiently from water. Fish RBCs are also more elliptical in shape than mammalian RBCs, which are biconcave discs. Additionally, the number and size of RBCs in fish can vary depending on the species, the water temperature, and other factors. Overall, RBCs in fish play a crucial role in oxygen transport, helping to maintain their health and survival. In vertebrates, the hemoglobin molecule includes 4 globin chains that create a stable tetramer formed by 2 alpha-like and 2 beta-like chains with 141 and 146 amino acid residues, respectively. Each one contains a heme group, which allows the binding of 4 oxygen molecules. The main function of hemoglobin is to transport oxygen from the gas exchange organs to peripheral tissues. It must be able to bind oxygen strongly but at the same time to release it when necessary, depending on the partial pressure of the gas. Reversible oxygen binding is possible thanks to the heme group, specifically with the participation of an iron atom in the ferrous form for some species of primitive fish such as lampreys and hagfish. Reversible hemoglobin dissociation occurs in response to oxygen binding. In the case of lampreys, the oxygenated form is monomeric, undergoing associations with dimers and tetramers when. In hagfish there are 3 monomeric hemoglobins in the oxygenated form, but when they release oxygen molecule, there is an association to form heterodimers and heterotetramers. White blood cells, also known as leukocytes, are a type of blood cell that plays a critical role in the defense against pathogens. They make up a smaller percentage of the blood volume than red blood cells, with a normal range of 4,500 to 11,000 WBCs per microliter of blood in humans. There are 5 types of WBCs: neutrophils, eosinophils, basophils, lymphocytes, and monocytes. Each type of WBC has a different function in the immune response. For example, neutrophils are the most abundant WBC and play a role in phagocytosis, while lymphocytes are responsible for producing antibodies and fighting viral infection. Neutrophils, they are the most abundant type of WBC in fish and play a role in phagocytosis, the process by which they engulf and destroy pathogens. They are the first to arrive at site of inflammation known as leukocytosis. Their functions include phagocytic, chemotactic, bactericidal. Eosinophils attack parasites too large to be phagocytosed and kill them. Basophils and mast cells have similar morphology. Basophils are found in circulation while mast cells in connective tissues. Macrophages are phagocytic cells, meaning that they engulf and digest foreign substances, including bacteria, viruses, and other pathogens. They also play a role in antigen presentation, which is the process by which the immune system recognizes and responds to specific pathogens. Macrophages digest foreign substances and then present pieces of the pathogens, called antigens, on their cell surface. These antigens can then be recognized by other immune cells, such as T cells and B cells, which mount a specific immune response against the pathogen. In fish, the specialized blood cells that are involved in blood clotting are called thrombocytes, which are similar to platelets in mammals. Thrombocytes in fish play a critical role in hemostasis, which is the process by which bleeding is stopped after an injury. In this slide, I have mentioned about the complete blood profile of some commercially important fishes analyzed through automated hematological analyzer. Blood collection from fish can be done in several ways, depending on the size and species of the fish, as well as the purpose of the blood collection. Here are some common methods for collecting blood from fish. Regardless of the method used, it is important to handle the fish gently and to minimize stress during the blood collection process. In addition, it is important to use sterile equipment and techniques to minimize the risk of infection or contamination. Thank you and keep watching.